Zebra mussels, microplastics from wastewater, E. coli concerns, and toxic algal blooms. These are all problems facing Lake Winnipeg that we've long reported on. But now a lesser known part of the story is actually getting some close attention, and it could shed light on the future of Manitoba's largest lake. Emily Chase is a microbiologist, a virologist, and a professor in the Department of Biology over at the University of Winnipeg. She's the first person to study algal viruses in Lake Winnipeg. Her work will contribute to building accurate climate change models for the lake, and she's in studio to explain. Good morning.
Good morning. Thank you for having me.
So what's an algal virus?
So basically, they're similar to the viruses that infect us, but the major difference is microalgae are just a single cell. So if you can imagine, if you're infected with viruses and you're just one single cell, it's like the equivalent of taking over your whole body instead of just some cells in your body. So if you're infected with a virus and you're an algae, it changes your whole lifestyle.
And then does that go on to affect the fish who might eat it and down the food chain, or how does it go on to affect the water, the lake?
I would say the biggest thing is actually kind of positive, in fact. So when you have microalgae blooming and you have a really dense population, if there's a lot of viruses infecting those, it can help collapse the bloom. So this could be positive for fish and fish stocks, and then positive for us as well.
Are there any drawbacks or negatives that you're looking at as well?
I haven't ever heard of something that would give like a negative impact on humans. It's perfectly safe to swim in the lake when there's lots of viruses. So mostly overall, they're pretty positive for us, in fact.
Interesting. So tell me a little bit more about the kind of work you're doing to study this. What have you been doing to study it?
So I'm lucky enough to be able to go out on the lake and sample aboard the motor vessel Nemeo. And that's with the Lake Winnipeg Research Consortium. And so I get to go out there and collect water samples, bring them back to my lab. My trainees get to filter those water samples and isolate viruses. And then we do what's called metagenomic sequencing. So it's a big process, but in the end, we get to figure out who's there, what kind of viruses, what kind of microbes are they impacting, and what their behaviors are.
How does all of this connect to building accurate climate change models for the lake?
So we're kind of a missing piece to the puzzle. There's lots of experts looking at phosphorus, nitrogen, other nutrients in the lake, and this is going to impact the amount of algae that are going to be there. But we kind of skip a step and we go to, what toxins are there from the algae, how it's impacting fish, and on a greater scale, how that affects us traditionally, recreationally, economically. But we're missing the step that occurs at the microalgal level that helps kind of collapse blooms, and that is the viruses. So even if there's a lot of nutrients there and those microalgae could bloom because they have lots of food to eat, if the viruses come along and collapse the bloom, it's a totally different picture than whether or not nutrients are there.
Okay, so silly question, but maybe not. Can we add virus to the lake? Because if you never have algal blooms being the problem in the first place, and virus can burst these blooms, I think was your language, is that one of the things that your work might lead us to?
A lot of people consider that, and it's a really smart idea. But the fact is, it takes a long time to propagate viruses, and to get them to that level, it might be difficult. But it is something that people should explore and something that we could think about, because you could effectively inoculate a bloom with these viruses. But yeah, stay tuned for that and see how that develops.
You've conducted research around Canada and all around the world as well. What stood out to you about Lake Winnipeg when you got into the research here locally?
There's so much excitement and love towards Lake Winnipeg. It's really important to Manitobans, and it's become very important to me for the same reasons. But it's also exciting as a virologist to enter like a new frontier, somewhere where like the research is untapped and we don't know who's there and what they're doing. So it's kind of twofold for me. I really want to contribute to the problems and help us understand the lake, but I'm also just very curious about this new frontier of viruses to look at.
What was it like when you first started realizing this and seeing that this data actually could lead to something bigger?
It was exciting, of course, and I knew that viruses had this sort of impact before coming here. It was just mostly excitement.
What are you and your team going to be doing in the future? What are your next steps then?
So right now we have people in the lab. I have students in the lab working on getting our data, and then we'll be right back out into the field once the ice is gone, getting samples in the spring. We get the samples in the summer and the autumn. And so it's kind of a threefold approach. We have people out in the field, we have people in the lab, and then we have people also on computers doing bioinformatics is what we call it, to try to figure out who's there, what viruses there are, and also what they're doing.
Pretty nice place to work.
Oh yeah, it's fantastic. I feel very, very lucky. Yeah.
Thank you very much for coming in and telling us about your work
Of
and its
course.
impact on the health of our lake.
Yeah, thank you so much.
That's Emily Chase with us live on CBC. She is a microbiologist, as mentioned, and professor in the Department of Biology over at the University of Winnipeg.